So transcription, the second chunk. So the first chunk was the central dogma. It was kind of an overview of everything put together. Really at the end of the central dogma, if we were in class, I wouldn't move directly to this transcription. I would continue with the whole idea of taking DNA to RNA to protein. And we work through the process. But it's actually a different kind of skill. It's a skill like more like a math skill or a logical skill. And so I want to actually continue through with the knowledge of what transcription and translation looks like from a molecular aspect, work through those lessons, and then as a separate piece at the end, talk about the skill of actually transcribing and translating, because that is something that we would expect you to be able to do. And if you decide to take biology to the next level, then you absolutely would be doing that as well. This learning that we're doing right here is often seen in second year in many schools. That's where you often take genetics. Often in first year it's more cell bio, but different universities have different kind of programs and flows. So this is where the lesson could diverge. So I'm doing the lesson slightly differently here online than I would be doing in class. For this lesson, like I do in class though sometimes, I have a PowerPoint, and the PowerPoint I'm not going to use. I'm going to use my picture point. So having the two up at the same time might be a valuable experience. So transcription involves the copying of the DNA gene from the DNA to an RNA. DNA is double-stranded. One of those strands is going to be the location of the gene. If I take a look at this graphic that I have here, so all these graphics are from your textbook and a variety of textbooks. So the transcription process, like replication, proceeds in several steps. There's an initiation stage that starts the whole thing. Then there's the elongation, where the process of the RNA is being created, and then there's a termination where it ends. And the same thing happens with translation. So the process of starting is actually pretty sophisticated, and as once again with the grade 12, we're going to simplify that. But remember, your DNA double strand got two strands, and only one of them has a gene. The gene can be located on either the bottom or the top. It depends on what gene you're looking for. Our DNA is incredibly complex, like our entire body. So in order to transcribe, the DNA helix must be opened up, just like we saw with replication. But in replication, it gets open and it stays open as it creates two things. Here, it opens up the site of where the gene is, it transcribes that gene, and it closes back up. So it unwinds, unwinds, transcribes, and then closes back up. Okay, that's the process. And the RNA polymerase is going to be responsible for this task. So when we look at the DNA has a whole bunch of things going on. But ultimately, the DNA is transcribed into an RNA, and that in turn is translated into a protein. Okay? So I'm going to draw the DNA here, and you'll notice that all of a sudden there's a red circle because I did do a quick edit here. So I'm going to draw the DNA, and I'm going to identify this side as my 3' and this side as my 5'. So if I was reading this, it would be from right to left. And on this DNA, you're going to find a bunch of things that are important for the overall transcription translation. Of course, there's the gene, and that's what we consider important. This is the actual area that's going to be transcribed and translated, most of it, transcribed and translated into a protein. So that's our gene. But in addition to our gene, there's a number of other things that have to happen. So the RNA polymerase itself, this is the enzyme that's going to polymerize the mRNA strand. It needs to bind to the DNA and do its thing. So it needs some help. So let me just talk about the region structurally, and then I'll explain how the whole thing works. In addition to the gene, upstream of where the RNA polymerase is going to bind, there's this thing. It's called the promoter. The promoter is a whole region, and the promoter region is responsible for yellow? Yeah, yellow. it's responsible, I'm going to make it nice and thick. Excuse me, sorry. I sneezed. I need to edit that out. Hopefully I see that and take it out. Otherwise, I'll just look silly. All right. And so this promoter sequence, what happens is there's a very specific and conserved sequence we find on most organisms. And this represents the promoter region. One is called the Tata box and one's called the Cap box. So collectively, these are both a part of the promoter. And this is where the RNA polymerase will bond. In between this Tata box and the gene, there's a region here. We're going to call it a UTR. Oops, make it nice and thick. UTR. There's one here, and there's one down here. And these regions, what they do is they are not translated. We call them UTR. UTR for untranslated region, because this one is at the 3' end. I know it doesn't make a lot of sense because of the way I've labeled this, but because of this is going to be at the 3' end of the mRNA that's created, this is called the 3', and this is called the 5'. Okay. Now, at the very end, there's another little sequence here. Oops. Another little sequence here that's going to code for something else that's very special. We'll talk about that in the next slide. Right, so I'll just write them down here so it makes some more sense. It's called a poly A tail. Now, this blue is the gene. It's the most important thing, and it's what's going to be transcribed and translated. So at the very beginning of the gene, always, you're going to see a special sequence right here, and that is going to be ATG. And there's a reason. You'll see why a little bit later. And at the end, there's always going to be a sequence you're going to see. Well, it could be one of several. Tag might be one of them. There's 2 other possibilities as well. So these are sequences you'll always see. So recognize this is a strand of nucleotides. This is the DNA. And there's a whole bunch of letters. And typically, they're in triplets that are going to represent the codons that are going to be translated into amino acids. Okay? But there are certain conserved sequences that let us know that certain things are happening, like the promoter and the bonding of the RNA polymerase. So this is the basic structure of the gene. So the RNA polymerase can bond, and off it moves. And recognize when this RNA polymerase moves along, whenever we create nucleic acids, they're always created in a certain direction. They're always created in a 5 to 3 direction. So as this thing moves here along the 3 to 5 strand, the new color I've not used, oh, I need a brand new color. So I've not used purple. Okay. So a new strand that's going to be created is going to be formed. And as it gets formed, this end here will be the 5' end because it's anti-parallel, and this end here, when it gets created, will be the 3' end. And I didn't do a very faithful job on this here because this won't actually be translated, right? Because it's the promoter. The translation is going to start where the ATG is. So this represents the structure of the DNA, and this is something you should understand as we move into the next step. Okay. So when the DNA is ready to be transcribed, the first thing that has to happen is you have to open it up, right, and expose the promoter and the rest of the gene to the RNA polymerase so it can do its job. And let me just go back and grab my RNA polymerase here. This program, when you switch and make a lot of different characters, it tends to lag a bit. And so the RNA polymerase will then bond to the appropriate strand. Notice there's my ATG, so this is my appropriate strand, and then it's going to move down. So that's 3 to 5, so the strand it's going to create is going to be 5 to 3. There's my ATG right here. So when we take a look at the process, remember there's initiation. So initiation is the first thing. So now let's go into initiation proper. So when you look at my PowerPoint slide, you'll see that I've given you details of what's happening during the initiation. I've also gone over in detail. Let me just pull the PowerPoint over here so you can see. I know I should have it at the same time on a different screen, but in case I don't, here's the information that you're using to get that understanding. Right? I'm just explaining it and you're following along with the PowerPoint. So what's going to happen is this right here represents my data box. And there's these things before the RNA polymerase can bond. So here's my RNA polymerase. Before it can bond, it needs a bunch of help. And we're going to talk about a few of these things in homeostasis. But there's these special proteins called, or molecules called transcription factors. And they bond first. So this will come to the data box and bond, this transcription factor. And then other transcription factors will come along. And other transcription factors will come along and they'll bond to the RNA polymerase in certain ways. So these are all these little things here, that, apart from the blue, are transcription factors. And along it comes with other transcription factors that are helping. And more transcription factors. And this whole thing, now the RNA polymerase is aided thanks to the transcription factors and is able to bind to the DNA successfully with high efficiency. Once the transcription factors have aided the RNA polymerase binding, then typically energy is expended in the form of ATP. The transcription factors will then leave because they've done their job, right? Well done. And the RNA polymerase is now free to do its job. Okay, so let's see if I can flip this. Or no, I can't. I don't know how to flip it. So now it's able to move down the DNA and transcribe by the addition of these nucleotides and produce an mRNA. And of this nucleic acid, mRNA, and this will and then this will elongate. Right, so the whole process will elongate, and eventually it will produce a strand. Eventually your RNA polymerase will reach a termination point, and the termination point is really interesting. At some point we know that there is a sequence that is T, 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 A, T, T. There's some sequence. And when the RNA polymerase hits that, we know at some point, anywhere from 1,000 to 2,000 nucleotides, I know how vague is that this RNA polymerase is going to now dissociate. And now we have the original DNA, which will reform back into its double helix. Oops, I made a mistake here. So reform back into this double helix, and then we'll have our mRNA, which is free to do the next step, whatever the next step is. So there's still more. It doesn't leave the nucleus yet. There's still something else that has to happen. Okay. So once the mRNA is created, so that's what this is now. This is not mRNA. This is the first RNA. So we call it the primary transcript. Yeah. And so we call this HN. RNA. Okay, HN stands for heterogeneous nuclear. We also sometimes will call it the primary RNA transcript. So many different things, so many different ways to describe the same thing. Okay, so this is the very first transcript that's been transcribed from the DNA, but this is not the final version yet. I have this video that you guys can watch. I encourage you to watch it. It shows the whole process with a nice little animation. I'm not going to show it here. I'm going to keep on working through the process. So the RNA strand needs to be processed. And the processing occurs inside the nucleus. So once the hnRNA is created, it needs to be modified before it becomes the fully mature RNA. So what does that look like? So let's pretend that this thing here that I've created here is my mRNA. And my mRNA is made up of several different segments. I'm going to label them, right? So I'm going to call this, I'll go with red here, or purple, whatever this is. This is A, B, C, D, and E. Okay? So I got these different segments. And what actually happens is, when you look at the DNA, we recognize that some of the DNA is expressed and some of the DNA is not. So let's say that A will be an expressed region. Let's say that C will be an expressed region. So it's going to be expressed, it's going to be transcribed and translated into a protein. And then E is also the same thing. And let's say that B and D are intervening regions. So we have acronyms. Expressed regions are called exons, and intervening regions that are in between the expressed regions are called introns. Okay? So what's going to happen is the exons are going to be stitched together, or the introns are going to be taken out. So this region here oops, I didn't get the whole thing. Let's try that again. This region here will be stitched, will be taken out. This region here will not be used. This region here will be taken out. This region here will not be used. And this region here will be taken out. So these three regions here, the exons, all get stitched together. So now my sequence is going to read A, C, E. And the B and D parts are junk. Right? They get broken down, that dark DNA world, maybe that something happens to it that we don't understand, but for now it's broken apart and then reused to build new DNA or RNA. So B and D are gone. So this is a spliced version, but it's not complete. We're not fully processed. So let's say that this is the 5' end and that this is the 3' end. So on the 5' end, something else gets added, and what gets added is this 5' cap. It's a guanine cap. It's a special nucleotide that goes on the end, and it helps protect it. And on the other end here, you might remember on the other one I said I put a whole bunch of A's. So on the other end, we have this thing called the poly A tail, and that gets added on this side. And so the cap, it allows the mRNA to be recognized by the ribosomes and protects it so that it doesn't degrade. So it's like putting a big, protection on the end of a hockey stick when you tape up the end. And the poly A tail also protects it from degradation and allows the RNA to leave the nucleus. It's a signal. It's like, this is the key to leave the door, the nuclear membrane. Okay. So now this entire thing that I have here, let's move my exon. Okay. So this entire thing here, this represents my mRNA. It's my mature RNA. And this is now ready to leave the nucleus. So just recapping the process here, I have my DNA. My DNA is initially transcribed into this hnRNA. And then this gets processed into mRNA. And the mRNA is the final transcript that's going to leave the nucleus and go into the cytoplasm. And the DNA, nothing happens to it. It just closes back up, and the next time that gene's needed, it'll be transcribed again. Right? So the RNA polymerase does this initial hnRNA, and a bunch of other enzymes and molecules will do the processing, and you end up with this mature RNA. And that's how the transcription process proceeds. Okay. So you can review the PowerPoint to get those points. I'm going to end this here, and I'm going to do the next chunk as translation.